The other type of in vitro method is a product study. Here you are analyzing the chemical composition of the cells to answer some question. Product studies can be qualitative or quantitative. Qualitative product studies ask some question about the structure of the molecules in the cell. Quantitative product studies are interested in the concentrations of particular chemicals in the cell. These experiments can be endpoint assays, or they may contain multiple time points. In all cases, a product study involves first purifying the component of interest from the cells and then characterizing the purified material using some appropriate analytical instrument. Let's start with an example. The Kiesling lab in Amherst performed a well-known project in which they optimized production of amorphidine, a precursor to the anti-malarial drug artemisinin. Their experiment involved variant biosynthetic pathways and strain modifications, and in each case they need to quantify how much chemical each strain produces. In this chart, they are performing an endpoint product study. For the best strain available in each year, they are quantifying the maximal yield of amorphidine in the cultures using GCMS. For such an experiment, they needed to choose an appropriate extraction method and an appropriate analytical method for detecting the molecule they care about, amorphidine. There are many methods for extracting small molecules from cells, and each isolates a different subset of the material. There are very general methods like methanol extraction, toluenization, hot phenol, and detergents, but there are also protocols that more specifically isolate a particular fraction of the cell. A Bly-Dyer extraction procedure is used to isolate membrane lipids. For alkaloids, there are ammonium extraction methods that take advantage of the distinctive pH-dependent solubility of these molecules. The best method for any molecule is something an experienced analytical chemist can determine, but it does not reduce to trivial rules. There are also many choices for analyzing the product. In liquid chromatography, the mobile phase is a liquid, typically water, methanol, or acetonitrile, and the column is beads, typically coated with C18 hydrocarbon chains. Usually the tail end of the liquid chromatography instrument is connected to a mass spec. LCMS is the method of choice for non-volatile chemicals. GC involves a gaseous mobile phase and is useful for volatile chemicals. It is also usually connected to a mass spec. You typically have more than one detector on any chromatographic instrument, and UV is very common and can be used to quantify the amount of the material in the sample. For some chemicals, including radioactive ones, thin layer chromatography is preferred. In addition to chromatographic methods, some product studies will require closer inspection of the molecule structure. For this, the various NMR techniques can precisely identify the structure of a purified molecule. In the amorphidine example, the extraction method chosen involves solubilization with methanol, followed by extraction in ethyl acetate doped with an internal standard, in this case trans-caryophyllene. The internal standard is used to calibrate the concentration of the chemical in the sample. If you know the molar concentration of the standard, you can relate the intensity of the peaks to this value and back calculate the concentration of your unknown. For the analysis step, they choose GCMS because amorphidine is mostly hydrophobic and thus volatile. They get traces off the column, and using authentic standards they can determine at which time amorphidine and caryophyllene elute. Thus, they can identify the peaks of these compounds in their spectra, integrate the area under those peaks, and use those numbers to calculate the concentrations. By comparing different strain designs, they can identify modifications that improve the yield by doing this assay on each one. In this example, we are performing an endpoint analysis on a single chemical component. A related technique is metabolomics. Here you are performing an endpoint product study, but examining many different metabolites in the cell. Experimentally, the basic setup is the same, but it requires a more expensive, higher resolution and lower speed mass spectrometer. In other related experiments, they perform time courses. Here they are following a culture of amorphidine-producing cells in a bioreactor over time and quantifying the optical density, the amount of amorphidine, and the amount of acetate at different times. In the previous endpoint experiment, the goal was to compare two different strains. In this experiment, the goal is to characterize a single strain in the context of a scaled-up bioreactor by varying the different conditions for the fermentation. They want to know how to optimally run the bioreactor to maximize product yield. Acetate accumulation is a common byproduct during fermentation and is one route by which carbon can be lost during conversion from glucose. Minimizing this loss is often the goal of these projects. So when dealing with microbial chemical factory apps involving small molecules, the scope of analytical techniques you need to consider is fairly straightforward. When you diverge from the small molecule apps, things get more complicated. Here is an example of a microbial chemical factory app that produces a more complex product. It makes a protein containing an alpha hydroxy linkage in the backbone. Thus, there is an ester in the peptide backbone. The E. coli cell is engineered with 3 genes. The first encodes the target protein. The site where an alpha hydroxy acid will be incorporated is encoded by a TAG stop codon. The cell also contains a tRNA that can decode the TAG stop. The third gene is an aminoacyl tRNA synthetase that specifically charges the tRNA with the alpha hydroxy acid. There is a long story to how you engineer such synthetases and tRNAs, but the question for us today is how do you analyze the product here and determine whether this thing works? Like any product study, the first step is to purify the component you wish to analyze. Because the target is a protein and we have control over its gene sequence, we can add a his tag to it and purify it by nickel NTA chromatography. Just like with the small molecules, there are mass spec methods for detecting protein products. The LC-MS setups used to monitor small molecules typically don't work for larger proteins. It's a more expensive mass spectrometer for proteins, but the basic idea is the same. You will ionize the protein and zip it through a magnet. In electrospray mass spec, you vaporize the sample, and as the solvent evaporates, ions get trapped in the particle, giving it charge. This allows detection by mass spec. You end up with several bands in the spectrum for each protein corresponding to particles with different charge. Software can back calculate the size of the protein from this data very precisely. In the case of the alpha hydroxy acid, the only difference between the desired product and the normal all-peptide product is an exchange of a nitrogen atom for an oxygen atom. Thus, the size difference turns out to be only 1 Dalton. However, electrospray is sufficiently precise to distinguish this subtle difference. For this particular product, there is another analytical method we can use to unambiguously confirm the identity of the product. Unlike peptide linkages, an ester linkage is susceptible to hydrolysis with base. So if you incubate the sample and some controls in sodium hydroxide, the desired product will get cleaved at the site of the alpha hydroxy acid. You then run a page gel to separate the fragments. In lane 4 of this gel, you see the expected fragmentation of the ester-containing product. What we've been discussing is analytical chemistry, and there's a great textbook covering all aspects of this field called Principles of Analytical Chemistry. In the real world, your choices about the appropriate test protocol will begin with a literature search. You will locate papers where the experimentalists analyze molecules similar to yours, and then look in the materials and methods section for how they did it.